I start here with a small picture of an analog soil systems modeling that was actually the farewell present of my former colleagues at the University of Hohenheim, where I was before. And this symbolizes quite well what is really in the focus of my research, and that's soil microbial mechanics that you can see here. So I'm very interested in in modeling how microbes transform organic material in soil, so in a much broader sense. And yeah, I start to, of course, the work that I present here was not done by me alone, but I have to thank a number of team colleagues and also collaborators, which are listed here. And I'm quite sure that I forgot also the one or the other, and I will not read all the names. So I have two slides on the motivation. So the first is, it's clear that soils are key to addressing global challenges, that it is symbolized here because all the soil functions or the functions that soils provide directly control a number of ecosystem services, which are then the underlying control of the challenges that we face and that are expressed in the 17 Sustainable Development Goals. And soils can make a contribution, so soil science can make a contribution to all of the 17 goals, sometimes in a quite direct way and often in indirect connections. So from the perspective of soil systems modeling, to make that link from soil functions to the Sustainable Development Goals and ecosystem services, we have to understand the basic processes in soil, biological processes, physical processes, geochemical processes, and they control supporting and degradation processes of soils, which then determine the states and properties of soil, which is depicted here as a soil natural capital. And the states and properties of soil, they feed back to the basic processes because they are dependent on the states in soil and the properties. And finally, the states or the soil natural capital determines the ecosystem services, regulating services like climate regulation, buffering and filtering services, but also provisioning services for biomass production and the use of soil and the function of soil as a habitat. Okay, so the essence of soil systems modeling, as I see it, is that we want to understand, quantify, and predict processes in soil. And we have some challenges coming with these goals. And the first one, and I mentioned a few here that I try to address a bit in my talk. And the first is that we have still fundamental knowledge gaps, and that's specifically true for biological controls of transformation processes in soil, but also structure formation of soil, for example. Then we have knowledge gaps related to the resilience of soil systems to stress, and especially to multiple stressors. Then when it comes to data model fusion, we have a challenge in getting the right information content of measurement to match it with models. And that brings me to the next challenge here that we have to address is the integration of knowledge with models. And last but not least, concerning the prediction goal here, we have to address the challenge that we need fast predictions that are reliable, and process-based models are often too slow to directly provide real-time predictions. Okay, I want to address these challenges related to three topics that I will address here in my talk. And the first one is transformation of agrochemicals, and this is mainly based on work done in the Collaborative Research Center campus, which was coordinated by the University of Tübingen. And then I will talk a bit on soil organic matter cycling. This is related to work done in two DFG funded, in one DFG funded project, the priority program Soil Systems, which are running in the first phase currently, and the next phase, the second phase starts next year. And the other project is the crop project. Both of these projects are partner projects of FINOROP. And then in the last part, I will talk more about a vision of how we could achieve the digital twinning of soil systems in order to address the last challenge that I mentioned before, to get fast and reliable predictions. Okay, let's start with the transformation of agrochemicals. But before I do that, I want to introduce a basic concept in soil science. So my work is mainly, or our work is mainly focusing on predicting processes at the pedon scale. So what do we call a pedon? The pedon has been defined here in this book, general lecture book on soil science, as a smaller spatial unit of soil cover. And it's typically envisioned as a 1 meter by 1 meter polygon, as you can see it here. And that's a unit of soil where the properties are more or less stable spatially in spatial respect. Okay, so the overarching goal of the CRC campus was to analyze pollutant and agrochemical fate at the landscape scale, and I was involved here in the sub-project P6 soils. And here one of our questions was to address the question, why do intrinsically biodegradable pesticides persist in soil? And we wanted to quantify the role of soils as sink or secondary sources of pollutants, and study the sensitivity of microbial pesticide degradation to specific habitat conditions, and use models, gene-informed models, and I will explain in a minute what that is, to identify relevant processes that need to be considered in upscaling, for upscaling this to the landscape scale. So in campus, we developed gene-informed models for pesticide degradation to answer the question if, or to analyze the question if pesticide degradation is limited by gene regulation. And this is depicted in this sketch here. So we consider the production of functional genes and gene transcript. And actually this step here from the translation step from the gene transcripts to the enzyme, we did not consider explicitly in the model, but assumed that this is directly proportional for simplification reasons. And then we used such a gene, and the advantage of such a modeling approach is that we can inform these models with measurements with molecular measurements of gene abundances and gene transcripts. And here you can see a typical outcome of such a modeling exercise. So what we simulated here was a lab experiment where a 14C labeled pesticide was added to soil, and then the soil was incubated in the lab at controlled condition, and then we measured the 14C mineralization so which is a measure of the degradation of the pesticide. And you can see here the blue dots are the measurements, and the lines are different model variants. So and this V0 variant is a model variant with the most complex representation of processes, and I will not go into the details but the other model variants, so the higher the number, the less complex the model structure. And you can see that only that the model structure with the highest complexity fits the data best, but also the slightly simplified version V3 here does also not perform that bad with respect to the mineralization. But when we also are able to predict the gene transcripts of a functional gene that encodes for an enzyme that catalyzes the degradation of that pesticide. And we can see here that the model is able to predict that, but in the more simpler modeling variant, which is more so this V4 variant is more or less a quite standard modeling approach that is normally used, that cannot readily capture this, especially the second peak, but also not the first peak very well of the gene transcript abundance. And we can also then measure the total gene abundance, which could be seen as a measure of the total biomass of the microbes. And here the most complex model variant performs best, actually. Okay, we can then go a step further and also analyze the relationship between mineralization rates as predicted by the model and transcript, either measured or modeled. And what we can see here in the upper left corner, this is based on the measurements, so the rates, mineralization rates extrapolated from the measurement against the measured transcripts. And there you can see that we have an historetic relationship. And this can be also reflected, especially by our most complex model, but also to some extent by the more simpler model. But you see also, so the upper and the lower row are different parameterizations. And there are some parameterization which fit the mineralization quite well, which don't show this historetic behavior, which we would expect according to the data. And this hysteresis in this relationship is important because there have been several claims that this transcript copy numbers can be directly used to derive mineralization rates. And as we have this hysteresis, it really depends at what time point in the mineralization kinetics we are. And this was shown by the study also. Okay, then we also looked at a different aspect of limitations that lead to pesticide persistence in soil, and that's the spatial control of pesticide degradation. And here we performed modeling study using the calibrated, a similar calibrated model as I have shown you before, and we analyzed what's the effect of different spatial localization of microbes in a soil pedon. So that's the scale that we look at here is around 50 centimeters width, I think 30 centimeter width and 1 meter depth. And we were interested if so what you can see is we have a homogeneous reference case where all the microorganisms are homogeneously distributed over the whole space here, and we had different heterogeneous cases where microbes were distributed heterogeneous. And we set up these heterogeneous distributions distributions using a spatial statistical model that was derived from measurements of bacterial cell distributions in real salts. And here you can see a short video what's for one of the heterogeneous cases. So on the left, you see the distribution of microbial degraders. So everything where it's yellow, we have high abundance of microbial degraders. And if you look carefully here in the videos, you can see that at these positions, the pesticide is degraded quite fast. Whereas in very dark blue positions, we have a longer persistence of the pesticide. And I show that here for two scenarios, for a continuous light rain scenario and for a heavy rain scenario where we have two precipitation events. And of course, with the higher precipitation events, we have a deeper penetration of pesticides into soil. But you can see this effect of higher persistence due to spatial heterogeneity. And for this pesticide that we looked at here, that's a pesticide that can be degraded quite fast. So we have not much of a difference in terms of leaching. So, but we have a much longer persistence in heterogeneous cases. Okay, with that, I come to soil organic matter turnover. And here we face a fundamental challenge because we experience a paradigm shift in the conceptualization of soil organic matter turnover in soil. So the old concept was a stable paradigm where theory, measurements, and models matched well together. And this old paradigm was relying on the theoretical concepts like chemical recalcitrance of substances, molecular condensation to the formation of humic substances. And we had models, one famous here is the Ross-C model, that enabled us to predict what we could measure relatively well. So at this stage, we had prognostic carbon turnover models. But there was a paradigm shift, as I said, and which considers much more the role of microbes and the accessibility of soil organic matter and saturation and other ecosystem controls. And this lead to a divergence in all of these three corners here. Because at the same time, we have also advanced analytical measurements techniques. So in the biological field, we have omics techniques. In the chemistry field, we can characterize the soil organic matter with quite advanced methods and analyze the composition of soil organic matter much better. Then there was in physics, we have quite good access to the spatial structure using computer tomography to visualize the soil pore network and so on. And also on the modeling side, there have been quite some developments towards more diagnostic and mechanistic models that are also spatially explicit. One example I've just shown you before, and that also considers for microbial traits. So, and that brings us to such a picture. Here you see the soil pedon from before again, and that's a quite complex view of what processes we have to consider if we talk about soil organic matter cycling. Don't go to the detail. The most important part here is that we have to consider interactions between plants and roots, and that we have to consider different complexity of soil organic carbon compounds and also different functional groups of microbes. And this microbe-centered view here is also visible here, so that breaks this more complex picture down with focus on the microbial turnover of soil organic matter. And more or less all carbon that comes into soil goes to the microbial community, and that to a large extent determines then what are the fluxes of CO2, what are the fluxes of energy as well, and how soil organic matter is stabilized. Okay, and we performed a systematic review to analyze the conceptualization of microbial traits and microbial life strategies. And here just the definition what a microbial trait is. So a microbial trait is a measurable microbial characteristics that are related to fitness and performance. And we systematically reviewed the literature with respect to soil organic matter cycling here, and categorized these 5 categories of traits that we have to consider. And I will not go into detail here, but this is important because it allows us to derive really measurable quantities that can be used to inform models at the end. That was the main goal of the systematic review. It's not yet finished. We are still in the process of data analysis. That's why I will not show too many details. So based on these functional traits, we can define microbial life strategies. And in principle, there are 2 types of concepts for that. 2 model ones, which you see here below, are and K selected, and which appear also on that picture here. And 3 model ones, which are taken mostly from plant ecology. And here on the right, you see the very recent refinement of one of the CSR strategy here. And here 3 functional types of microorganisms are considered: stress tolerators, competitors, and they are distinguished with respect to the limitation by resources. So competitors are good when the resource constraints are low. That means when the availability of resources is high. Their stress tolerators can cope with conditions where they are really limited in resources or stressed by further stressors. And then these 2 groups are distinguished from a 3rd group, the opportunist, and according to what they call here resource variability, and that is meant in a temporal context. So what is meant here is a temporal resource variability. Okay, we used a bimodal microbial life strategy scheme to parameterize a trait-based soil organic matter model that is depicted here. So we distinguish here between copiotrophs and oligotrophs, and they are parameterized according to the growth rate, their maintenance requirements, and their activity, their activation rates, so their switch from active to dormant state, and further functional microbial traits. And we did again a literature review to constrain the model, and tested that against a lab experiment, again a pulse labeling experiment where we placed 13C labeled litter on top of a soil core. And for the modeling, we only considered that first early phase litter, so very fresh organic carbon that was added to soil. And then we used a model data fusion scheme that was originally developed for hydrological science by Garari et al., and extended that by including a Markov chain Monte Carlo assembler to, at the end, get to representative parameter spaces. And this approach successively reduces the large parameter space of such process-based models step by step by applying parameter and process constraints. And we have to do that successfully because we are looking at the needle in the haystack. You can imagine that you have, we did that for a 60-dimensional parameter space. And of course, in a 60-dimensional space, you are looking for very small parts of that high dimensional parameter space really contains a constraint but uncalibrated parameter space. I cannot go into detail, but this approach is widely applicable to many biochemical models, and we applied it meanwhile also to several models. So what comes out here, what you see here by the lines, is a model prediction. And that's really a prediction. It's not a calibration to the data. That's why you have quite some high uncertainty bands. And on the top here, you see the litter-derived carbon in the microbial biomass. And as you can see, the model predicts that relatively well with a relative okay uncertainty. And we can also predict the litter-derived carbon in the total organic carbon. And when it comes to the specific functional groups here, we can see we have this high uncertainty, but that's due to the fact that we have low constraints to constrain these groups. Or we have weak constraints. But if we then predict the proportion of the remaining litter carbon, so how much of the litter input remains in soil, then we can see that we can predict that with a relative low uncertainty despite all this uncertainties here. So that gives us some hope that we can use such model data fusion approach to come to predictive forecasts of soil organic matter stabilization. Yeah, and I cannot go in the details here, but in the frame of the soil systems SPP, we extend this modeling approach here by also incorporating the energy flows that go with the mass flows, and this is further constrained by heat measurements. But due to time constraints, I cannot go into the details. So the latest version where we also apply this constraint-based model conditioning is for soil plant modeling with XPERT-N. That was one of the last projects I started when I was at the university in Hohenheim. And here we test this approach against a 9-year data set that characterizes the soil vegetation and land surface processes in two climatically contrasting regions. And we focus here on one of the regions. And the hypothesis is here that the constraint-based parameter sampling can improve the predictive power of the model compared to normal calibration. Okay, with that, I come to the crop model, to the crop project. Here, the basic idea is to combine two root contrasted phenotypes to come to more resilient agroecosystem, and the core idea is to just increase the soil volume that is accessible by plants to make them more resistant against drought, especially. And we have here an experimental cluster and a modeling cluster. And I will report here on that rhizosphere scale modeling a bit. So we have developed a rhizosphere scale model, which is 1D axis symmetric, that implements this trait-based model that I have shown before and coupled it with transport processes in the rhizosphere. So that means in a small region around such a root segment. So we are talking about a few millimeters here. And we also applied the process constraint calibration pipeline here. And we can see that here an example how the parameter distributions shown as a scatter plot of two parameters against each other change after applying only constraints related to parameters of the model and then parameter plus process constraints together. not in all, but in some cases, we can see that the parameter space gets reduced quite significantly. Okay, and that's an outcome of such a model. So what we can see is the spatial profile from the root into the soil of different organic carbon pools, so large complex carbon and dissolved organic carbon, more simple compounds, and we can see due to root oxidation that we get a spatial gradient of the substance from the root into the soil, which has a depth of a few millimeters, as you can see here. And the microbes react to this root exudation, and you can see that here for a bit magnified for the two functional groups. And the copiotrophs are fast-growing organisms. They show a much more narrow gradient from the root surface. So, and finally, this goal is of course the goal is of course to link this fine-scale model in the rhizosphere that simulates a root segment to a larger scale model. And for that, we are currently in the process of coupling it with the seed plant box model, a structural functional plant model, and also a macro-scale model to be at the end. And the vision is at the end that we can really predict first soil plant interactions from above ground to below ground at the pedon scale, but also then at the field scale. Okay, with that I come to the last part of my talk, which is the vision of how we can come to a digital twinning of a soil system. So that means representation of the real system, which allows to simulate the real system close to real time and inform the system also with real-time observation and monitoring data. And I think these constraint-based parameter sampling that I used here can be used to create mechanistic models that can that yield to plausible predictions, and then we can use these models to train and validate surrogate models, which run much faster. And at this stage, they could be informed by observation data. And these surrogate models can then be used for computational demanding sensitivity analysis and model reduction studies that we have done also in my previous research, which I could not show here in detail, to improve the mechanistic model. And in this circle, we come to a hybrid model to get the best of both worlds. Okay, and with that, I think such a digital twin of the soil system can then also contribute as a module of the digital agricultural avatar due to time constraints. And here I make some advertisements. We have currently an open position for a PhD position for hybrid soil microbiome modeling. And with that, I'm at the end, and sorry for the delay.